In this video we will discuss about some of the basics of glycosylation. So glycosylation is a multi-level regulation process. So we will start with the central dogma, that is DNA is converted into RNA. So at the transcription level also, this glycosylation can be regulated through various factors. Those factors could be epigenetic factors, that is change in the transcription level of the genes, or transcription factor that helps epigenetic factors, microRNA or RNA stability. So these all can change the transcription level of the genes. So next is the translation. At translation step, where RNA is converted into protein. So in this case also, in this step also, this glycosylation can be regulated through the various factors such as ribosome binding, microRNA, RNA binding proteins, or glycoenzyme degradation. If any of these factors could affect the glycosylation of their final product, as if there is no glycoenzyme, there will be no post-translation modification or glycosylation. Next one is the final step, that is during post-translation modification or glycosylation. In this case, the location of the glycoenzyme, the reaction rate, the Golgi transport or sugar nucleotide biosynthesis, glycoconjugate half-life, all this will affect this glycosylation. And these glycosylation are mainly occurred in the endoplasmic reticulum or Golgi apparatus. Finally, what will happen if any of the changes happen in any of the stages? It will affect its final function, it is molecular recognition, or it might trigger some undesired signaling pathway that is harmful for the cells. So what are these carbohydrates or that combine together to form this glycosylation? So there are total 9 carbohydrates that are found, 9 monosaccharides that are found in the humans. In this combination of these 9 monosaccharide will form a carbohydrate structure that are present on the protein scaffold or on the cell surface. This glycosphingolipids, these are the special class of glycolipids that are present on the cell surface. And for this O-glycan formation are usually happen on the serine and threonine amino acid of the protein scaffold, whereas N-glycan happen on the sequence, that is NXST, where N is asparagine, X could be any amino acid except proline. The next one should be serine and threonine. And this last figure is just the elaborate expansion of the first figure, in which it is mentioned that when the DNA, during this transcription of this DNA to RNA, the factors that could be affected is the transcription factors or epigenetic regulation, that is mentioned here also. And when the RNA is formed, this K degradation will also play roles, as RNA is not stable. It could be degraded if RNA is unstable. This K degradation will also play a role in the regulation of this glycosylation. Next step is, from RNA it will form first a protein scaffold or the enzyme that that will play a role for the glycosylation in later stage. So this protein scaffold will also might be degraded if the scaffold does not acquire its native structure. Simultaneously, this glycoenzyme might also degrade if they does not acquire their native structure. And if they acquire their native structure, they will move forward in the endoplasmic reticulum and Golgi, where these glycoenzymes resides, and they help in the post-translation modification or glycosylation of the scaffold, and they will finally form the glycoprotein. However, if this glycoprotein does not have their native structure, then it might also be degraded during any step in the endoplasmic reticulum or in the Golgi. So, and okay, so the final step is that this excess amount of glycoprotein might act as a feedback regulation that might control this transcription factor or epigenetic regulation in a way that the amount of glycoprotein remains uh, within a desired limit. So this is how multi-level regulation of glycosylation occurs, and how they control uh, each step, how each steps are interrelated in such a way that this glycosylation levels within the cells remains under control.